Good afternoon, guys. Hopefully you've enjoyed a little bit of this beautiful day out there so far. You guys are going to be delighted to know that today
Recording
is
in
a
progress.
short lecture. This is a smaller chapter, certainly by comparison to the other chapters that we've had. As you know, so far in the cardiovascular system, we talked about cardiac anatomy. We then had a lecture on cardiovascular physiology, where we even worked in a math equation to compute and arrive at cardiac output, that important measurable variable for cardiovascular health. And today we conclude the unit on the cardiovascular system with blood vessels. And as you guys know, we of course have already talked about blood vessels throughout the previous two lectures: arteries, veins, and capillaries. But today we'll take a little bit deeper of a dive in and specifically steer you in the direction of the content that you need to know to be successful on your lecture exam number 2. Next week, Tuesday and Thursday, we switch gears and we change to the respiratory system. So we'll learn about gas exchange and the lungs and the physiology and anatomy behind all of that. So that'll be next week, Tuesday and Thursday. And at the conclusion of next Thursday, you'll have all of your content toward lecture exam number 2. All right. So let's go ahead and get started. Today's going to look a little bit odd as far as your extra credit / attendance questions that you're going to send to me. Most of the time, there are four questions if it's a whole chapter, or 2 questions if we split it. Because this is a shorter chapter, there are only 3 questions today. So it's going to look a little odd. Don't think you missed anything. There are truly only 3. All right. All right, so let's talk about blood vessels. And here's something we'll start with that you guys have seen and you guys should certainly know inside and out by now. When blood vessels leave the heart, they are arteries with oxygenated blood. They're the largest in diameter, but fewest in number. And then they branch off and become arterioles, which have a smaller lumen, smaller inner opening diameter for blood carrying capacity, but there are more of them. And those arterioles, we're going to talk about a pretty good amount today. We're going to talk about how they are the major blood vessels that redistribute or redirect blood depending on what's needed. The arterioles then, in turn, branch off into capillaries. And the capillaries are by far the smallest in diameter of our blood vessels, but also by far the most plentiful, the most numerous. There are millions of capillaries, okay? Capillaries come into contact and touch all tissues, all organs, all muscles. All of them have to interact and be in contact with capillaries, because it's at the capillaries and the capillary beds where gas and nutrient, enzyme, hormones, all of the transfer of anything valuable out to cells, tissues, and organs occurs at the capillary level. Furthermore, the onloading of waste product like carbon dioxide that we've talked about over and over, and we'll certainly talk more about next week, as well as other metabolic waste products, are all sent to be eliminated from the body at the capillary level. Okay, then we start the return pathway to the heart. So the capillaries kind of take a reverse route on the venous side. They get less in number, but a little larger in capacity, diameter again of that lumen, the hollow, open, carrying portion of blood vessels. As they become venules, venules then become even less numerous and carry more blood at a given time when they become veins, and then veins, of course, return back to the heart, very specifically, and as you guys should be aware at this point, via the superior vena cava for everything coming from above the heart, from the inferior vena cava from all areas coming below the heart, and from the coronary sinus from those cardiac veins that are removing waste product from the heart muscle itself. Okay, all of that at this stage should be really, really comfortable, and you guys should know this by now. Know for the exam, It's underlined, so know this for exam number 2, that veins, very much like the lymphatic vessels, in fact, identical to the lymphatic vessels, other than the cardiovascular system is a closed loop and the lymphatic system isn't. But otherwise, blood being transported back to the heart from our veins has less pressure. Okay, that makes perfect sense if you think about it, right? Blood moves through the cardiovascular system because of hydrostatic pressure from the 4-chamber muscular heart. Okay, when that heart contracts and goes into systole, a systolic contraction, the blood that's ejected from the heart on the arterial side, because it's closer to the pump itself, has a lot more hydrostatic pressure. By the time blood makes its way through all of the arteries and arterioles and through the capillaries and is making its way back to the heart, on the venous side, in the veins, there's a significant dropout off in pressure because it's a lot further away from the heart itself. Okay, so the veins, much like the lymphatic vessels, rely on help from our skeletal muscles. When they contract and change shape, those muscles squeeze the veins and push blood along. Okay, and as we saw in lab, veins have valves for this very reason. Okay, oftentimes, especially from inferior aspects of the human body, everything below the pelvis, for example, not only is there less hydrostatic pressure because the veins are further removed from the pump, but they also are having to return blood against gravity, moving it back in a superior direction, fighting gravity on the way. So that's where that compression from skeletal muscles in combination with those valves that don't allow blood to go back downward all help flow and the ultimate return of blood to the heart on the vein side. All right, capillaries, just talked about this. All right, they are composed of simple squamous epithelial tissue. Okay, again, there are millions of capillaries. When we learned about tissues, we learned about those paper thin, fish scale like simple squamous epithelial tissue. They're paper thin, and anywhere where we have simple squamous epithelial tissue, the form meets function, meaning that these capillaries are designed and consist of only one layer of simple squamous epithelial tissue because it allows for osmosis and diffusion, the easy transfer of quality products from our plasma and our red blood cells, and then from the tissues and cells, the removal of waste through the capillary wall back into the plasma and erythrocytes. Okay, so again, form meets function, and the capillaries, while they're microscopic in many instances, are made up of the simple squamous epithelial tissue, all to facilitate the exchange of gases and nutrients and take on waste products with ease. All right, so what occurs within the capillaries? Okay, we've just mentioned, and you guys know by this point, gas exchange, right? When red blood cells make it out to the system, coming from the heart, the lungs, and via the arterial side, they're going to be oxygenated. And the oxygen molecules out at the cellular level are going to transfer, they're going to diffuse through those simple squamous tissues of the capillaries to the cells, organs, and tissues to provide oxygen, right? And of course, waste product of cellular activity includes many different cellular respiration type wastes, but predominantly carbon dioxide, which then will diffuse back into the blood and be removed back to the lungs. So when we exhale, now we can get rid of and eliminate that carbon dioxide. We'll inhale again, it will bring in new oxygen and the system will start all over again. Okay, so gas exchange is the huge product that I want you to know occurs within the capillaries and the capillary beds. Okay, when I say capillary beds, there are clusters of capillaries in given regions. We're going to talk later about the digestive system. There are capillary beds, clusters of capillaries that completely enclose and surround the small intestine. So when our stomach breaks down the food that we take in into the most basic form of carbohydrates, proteins, and fats, they're called monomers. They're microscopic versions of those nutrients. When they're that small, they can then be diffused from inside of that small intestine, pass through the intestinal wall into those surrounding capillary beds, into the bloodstream, and then our cardiovascular system then transports those carbs, proteins, and lipids all throughout the body as nutrients that we need to survive. Okay, so that's one instance of capillary beds. They're certainly found in other places. We'll be talking next week about capillary beds in the lungs for gas exchange with the alveoli of the lungs. All right. All right. The second thing that occurs, of course, with the capillaries that we previously discussed when we talked about the lymphatic system is lymph. Remember, the cardiovascular system is not 100% efficient. So when nutrients and fluids and gases are exchanged from capillaries to surrounding tissue out in the system, there is not 100% clean transfer. Okay. And as we talked about with the lymphatic system, that interstitial fluid, that fluid that isn't handed off cleanly from the capillaries to the cell, tends to acquire out there in that extracellular space until those lymphatic vessels pull it in and ultimately recycle it to the heart. Okay, so at the capillary beds, the two main events are gas and nutrient exchange and the formation of lymph, that extracellular fluid. All right, the structure of arteries and veins. We talked about capillaries already, and capillaries, remember, are simple squamous epithelial tissue. Single layer, very paper thin, allowing for a very easy transfer of materials through the walls of the capillary. Cardiac vessels, arteries, and veins, however, have three different layers, and we've talked about these last time in lab. Right? Tunica or tunic, meaning layer. We talked about this there, so I'm not going to go over this in great detail again, but the tunica intima is the internal most of the three layers. It has an innermost lining called an endothelium, and this is kind of a non-stick surface. I use the analogy, the comparison of Teflon in a frying pan. When our blood is flowing, especially those formed elements, our erythrocytes and our leukocytes and our platelets, we don't want them binding or clinging to the vessel wall. That's going to cause a clot or a clog, or at least slow down the movement of blood and valuable nutrients and gases. We want a smooth, slick surface that everything just passes through with ease. And that's what that endothelial layer, the innermost lining, and an interior layer of the tunica intima is all about. The tunica media is that muscle middle layer again. It is much thicker in the arteries than it is in the veins. And that is by design. Form meets function again. Our arteries, as we're going to talk about, are the major redistributing vessels. They're going to vasoconstrict if they want to steer blood away from an area. Okay, vasoconstrict, meaning this lumen, the hollow opening where blood is transported. During vasoconstriction, that lumen gets smaller in diameter, restricting or reducing the amount of blood traveling through that given arteriole. Okay, now there are going to be multiple instances where that occurs. One could be extreme cold, just as an example. You walk outside on a freezing cold morning, the arteries, your brachial artery in your arm may vasoconstrict to steer more blood back to your core, to keep your critical organs warm, and also to keep blood away from the surface area of your arms, your skin, so you don't unnecessarily lose heat. Okay, that's one example of vasoconstriction. Vasoconstriction also can occur during situations of trauma. If you really cut your forearm very badly and are bleeding, again, that brachial artery sensors will detect the injury. The tunica media in those arterioles will tighten. You'll have vasoconstriction, narrowing, greatly reducing the amount of blood flow to the affected area, thereby preserving your blood volume. Okay, those are some examples of vasoconstriction. Vasodilation is, of course, the exact opposite. This is where that tunica media, the middle muscle layer, dilates and it gets larger. It opens up the lumen so more blood flow can go. If I take off after our lecture for a run, when I leave out of my driveway, the muscles of my legs, those receptors are going to send a signal to my brain, which in turn is going to send a signal to my heart and my vessels, my arteries, specifically the arterioles, to vasodilate around my leg muscles, because those leg muscles need more glucose, they need more nutrients, they need more oxygen, so I can continue to perform and exercise without fatiguing. All right? So the arterioles, again, are the vessel type that are the most responsible. They by far have the largest responsibility for redirection of blood. By the way, the tunica media is composed of smooth muscle tissue that we talked about in Bio 210. Remember, skeletal muscle we can voluntarily control. We cannot voluntarily control smooth muscle. We don't consciously think and make our arteries dilate or constrict. It happens via the autonomic nervous system, and because of sensory input telling the brain that there's a problem, the brain in turn sending a signal to the arterioles, and then that tunica media adapts and changes sizes to correct for or adapt to whatever the situation may be. And then the tunica externa, that third outermost layer, is merely a protective coating, okay? This is going to be fibrotic, collagen-based, flexibility, but preventing tears or damage. So this is exactly what I just talked about in the last slide. So as you're studying, if you want to read that, you can, but that's exactly what I just said, so we're going to skip over this. All right, so the first of your three questions: which of these three vessel types permits nutrient and gas exchange between our blood and tissues? Where does gas exchange occur? All right, let's continue. All right, so this is something that maybe isn't real intuitive and may not make sense at first glance, but if we look at the proportion of blood in any vessel type or any aspect of the cardiovascular system at any given time, it's going to look a little bit strange. For example, the heart has about 10% of our blood at any given time. Okay, and this is, for the most part, what we're looking here number-wise is more of a resting situation. If we were exercising, that heart capacity would be a little bit higher. About 5% only in the capillaries. And that kind of surprises people a little bit because, again, there's millions of capillaries. However, they're microscopic. So the amount of blood within the capillaries themselves is extremely minimal, okay? In the whole pulmonary circuit, that blood vessel circuit from the pulmonary trunk to the pulmonary arteries to the lungs and back via the pulmonary veins, only about roughly, again, 10% of our blood is in that pulmonary circuit engaged in gas exchange. Okay, and then our arteries and arterioles out at the system are roughly about 15% of blood volume. So the disproportionate amount, the vast majority of blood at any given time is out in our veins and venules. Okay, so why? physiologically, why does that occur? We would kind of intuitively think that if it's a closed loop system, like plumbing in our house, if I turn on the water in my house, that water is going to be equally displaced through all the pipes. It's going to move all the way around. Okay. But there's two reasons why the bulk of our blood is in the veins and venules. And the first one goes back to the point that we've talked about previously, and that's the reduced hydrostatic pressure that is experienced over on the vein and venule side. Okay. If the artery is right near the heart where that pump is producing the greatest amount of pressure, that blood and the formed elements are going to move very rapidly because the pressure is significantly higher. By the time we get all the way out through the arteries and capillaries and into the veins, that pressure, again, is significantly less. And where there's less pressure, that blood then is going to move slower. Okay, there's less pressure, less push. That kind of makes sense, kind of, just from a common sense standpoint. The second reason, and the larger reason, why the majority of our blood pools in our veins and venules goes back to what I said about the thickness of the tunica media, that middle muscle layer. All right. This is where the form, the actual makeup of arteries versus veins comes into play. Okay. With the arteries, where that muscular, smooth muscle wall of the tunica media is much larger so that it can redistribute blood as needed, it can vasodilate and vasoconstrict, so it's much thicker. Therefore, the lumen of arteries is much thinner, smaller. Okay. Whereas over on the vein side, again, the tunica media is paper thin. It doesn't really dilate and constrict nearly the way arterioles do. So because this is a much thinner wall, there's a lot more lumen space. So if the vein is a lot larger, it can carry a significantly larger amount of blood. And because it can carry more blood in the lumens because of that diameter being larger, by default, the veins and venules are going to hold the vast majority of blood. Okay, so that's why. All right, tissue perfusion. We've talked about this before, right? Delivering oxygen and nutrients, removing waste from all tissue and cells. Some key areas where that occurs, and certainly next week, Tuesday and Thursday, we'll be talking about gas exchange within the lungs. Okay, those capillary beds that are intertwined inside of our lungs, meeting up with and touching the alveoli, offloading oxygen that we've inhaled onto the erythrocytes, and then offloading carbon dioxide off of the erythrocytes to the alveoli so we can exhale and remove it from the body. Gas exchange within the lungs, tissue perfusion. All right, within the digestive tract, we're going to talk about it later in the semester, but I gave you the example previously of how there are capillary beds that are intertwined and wrap all around that small intestine for the absorption of nutrients to carry to the body to be used. And then again, later in the semester, we'll be talking more about the urinary system and our kidneys and about the formation of urine. And by the way, when we get into that urinary system, some really interesting concept, because a lot of people think that our kidneys filter our blood and make urine. And while that is true, the real primary function, the vast majority of kidney function, is a recycling center. Our kidneys reabsorb and recycle 99% of everything they filter. Our kidneys only eliminate 1% as urine. So where people think the kidneys take everything from the blood and just ship it out as urine, that is absolutely not correct. Okay, they're tremendous recycling centers, and just a tiny bit of it gets eliminated as urine. But certainly there's a lot of capillary beds and all of those things that are, as I just mentioned, recycled. They make their way back into the bloodstream via tissue perfusion of the capillaries within the kidneys. All right, so this slide Guys, this looks like it would be a matching question because it was. 1 year ago, this was a matching question. I have taken this off of the exam. Okay. So that's why it remains highlighted and underlined and all that stuff. Don't spend a lot of time memorizing this. I told you there's only one matching type question from the cardiovascular system. And I told you before that that's that slide in the very first of the 3 lectures on the wall structure of the heart itself. Okay, the epicardium, endocardium, myocardium, and the serous membrane. Okay, all right, but what I do want you to remember for the exam is just the general concept of what's going on here, because I know there's at least one question on the exam about concept here of what we're talking about, I'm going to talk about here. You do not, I'll say it one more time, you do not need to memorize any of these numbers at all, okay? Just know the general concept of what's going on. So what we're seeing in these charts is a comparison of blood flow in a given region. Remember, I've been talking a lot about blood redirection or blood redistribution through vasoconstriction or vasodilation of arterioles, right? When some disruption or change occurs, and exercise is certainly a disruption or a change, blood needs to be redistributed to accommodate that change. All right. So what we're looking at here is blood flow at rest versus blood flow during strenuous exercise. All right. So the brain, our computer, our brain only requires oxygen, nutrients, waste removal at about an equal level, regardless of what we're doing. Okay. If we start exercising, our brain doesn't have an increased demand for oxygen, in other words. Okay. So it's pretty much steady state. It always needs the same amount of oxygen and nutrients. So the blood flow remains pretty consistent. However, in the heart, and as you guys already would guess and know, if we start to exercise and suddenly the muscles of our legs and arms require more oxygen and more glucose and more ions and enzymes like sodium and potassium, so those muscles can continue to depolarize and contract, we need more blood flow. So if we have more blood flow and that blood is going to get redirected from the arterioles, from our special senses, for example, or from our abdominal cavity, we're going to dump that blood into the system and our heart's going to see an increase in blood flow, getting that blood out to the areas that need it during strenuous activity. The biggest increase or redistribution, probably to no one's surprise, is going to be our skeletal muscles, because of course, when we're exercising, it is our skeletal muscles that are working. They're what are taxed. They need glucose. They need a lot more oxygen in order to not fail or become fatigued and exhausted. Okay, so no surprise that our heart and our skeletal muscles have an influx or an increase of blood supply during exercise. Our skin likewise gets a significant uptick of blood flow during exercise, and this has to do exclusively with maintaining homeostasis. If you're playing basketball outside in August in the afternoon, what's the very first thing that happens when you start to exercise? You start to sweat, right? So that is your blood flow redistributing your arterioles, distributing blood out to the surface of your body so that that plasma liquid portion can cool the body. Well, if you're losing plasma and water from your bloodstream, your blood pressure is in turn dropping, because blood pressure, as you guys know, is related to blood volume, and blood volume is for the most part water. So as you sweat water out and lose water, your blood pressure is going to diminish, okay? But this is the skin redistribution is all about homeostasis and temperature control. All right, now our kidneys, again, recycling centers, urine producers, okay? When we're at intense exercise and work, this is an area that can afford to lose some blood supply. recycling nutrients and forming urine and processing through what's good versus what's bad is not a priority at this time. That blood needs to come from somewhere. So the kidneys is a significant area where blood can be redistributed back to the skin, the muscles, and the heart. Okay, because at any given time, about 25% of our blood volume is in our kidneys. So that's a ripe opportunity for redistribution. Just like our abdominal cavity, organs like our spleen, our liver, our gallbladder, our stomach, digestion is not a priority while we're exercising. Okay, this blood can be redistributed. Okay, and then other really are going to be like our special senses, things like ears and lungs and things like that. Okay, those are areas where we can take blood from. So again, just understand for the exam the general concept here of what's going on, and that certain areas will lose blood volume to redirect it to areas of priority during intense exercise. And those big areas are kind of intuitive again: heart, muscles, and skin. Second of your quiz questions. So during vigorous exercise, what's going to happen? Will the capillaries of active muscles be engorged with blood? Will blood be rerouted and moved into the digestive type organs, or will our skin become cold and clammy? Should be a pretty easy one. All right, let's continue. All right, so blood pressure we talked about, and we talked about this at length in lab, okay? So I'm not going to talk about this in detail again here. We know that blood pressure is the pressure of our blood volume on the arterial walls, and that there are sensors, there are receptors embedded in the arterial walls. And systolic pressure is when the ventricles are in systole or contracting, in other words, and the diastolic pressure, that lower number, is obtained when our ventricles are in diastole, when they're relaxed. Okay, we talked a lot about cardiac output and how we derive that: heart rate times stroke volume. And if cardiac output increases and we have more blood being ejected per minute from the heart during intense exercise, for example, our blood pressure is going to increase because there's more blood in the arteries. More volume is going to be more pressure. Okay, if we get done exercising and we start to take a cool down walk, and our cardiac output starts to decrease and we have less blood pumping out of the heart, of course, our blood pressure in turn will also decrease. There'll be less blood volume, so there's less pressure on the arterial wall. All of that should make sense to you guys. All right, so again, talked about what systolic blood pressure is and what diastolic pressure is. Again, I told you guys that technically, yes, there's atrial systole and diastole, and there's ventricular systole and diastole, but globally, and unless you're in cardiology dealing with some type of a specific atrial defect or atrial disease process, for all other times in most of your careers, you'll never talk about that. You'll only be dealing with systole and diastole as the heart as a whole, so therefore referring to what those ventricles are doing, and that's because the ventricles are the workhorse of the heart. So we're talking about systolic blood pressure, we're talking about ventricular systole, and we're talking about diastolic blood pressure. We're talking about what are the ventricles readings going to be during ventricular diastole. All right, one thing we haven't talked about is resistance. All right, I mentioned earlier about the endothelium, the innermost lining of that tunica intima, the innermost layer of arteries and veins, and how it's desirable to have a smooth, slick surface so that blood can continue to flow with as least amount of effort as possible. The whole goal of the cardiovascular system is to move blood and all of the components that blood is transporting throughout the body. So the easier the heart can do that, the better. Now, resistance, specifically called peripheral resistance, because it's outside around the entire periphery, the system, is anything that slows down blood flow or makes it harder for blood to move throughout the arteries and veins. Okay, so there are three primary sources of peripheral resistance, things that can slow blood flow down. The first one is viscosity. Okay, viscosity is just simply thickness. Okay, you might have heard on commercials from time to time when you talk about motor oil, you hear about viscosity of oil. Okay, a 10W30 oil is a different thickness than 10W40 oil. Okay, that's the viscosity. All right, our blood can become thicker or thinner depending how much plasma and water. Remember, 60% of blood is plasma and 90% of plasma is water. So if we add water, plasma, we're going to thin out our blood. There's going to be more water and less of a percentage of the formed elements, the erythrocytes, leukocytes, and platelets. If we lose water from our plasma and our blood, when we sweat, for example, we're going to have a greater percentage of formed elements and less liquid. So then the viscosity, the thickness will increase, right? So that's what I want you to know here on this underlying portion for the exam is that if we have an increased viscosity of blood, thickness, there's going to be increased resistance. It's much harder for the heart to pump around formed elements than it is water, plasma, okay? With my cup, if I dump my water out, it has a paper thin viscosity. It's just going to pour instantly onto the floor, right? But if this cup is filled with molasses, which is very thick, it's going to pour out much slower. It has a thicker composition, increased viscosity. The second one is blood vessel length. And this will kind of make sense. The longer the vessel, the more resistance there is to move something through that vessel. Think if you had a straw and a little piece of wadded up paper in that straw, okay? If the straw is only 1 inch long, you can give it just a little bit of a blow and that paper is going to shoot right out of it, right? But if that straw was 10 feet long, you really have to have some force behind it to get it to move all the way through. That's just a longer course requires more energy. That's all it's saying. All right. And then the greatest influencer of blood resistance is what we've been talking about repeatedly through this lecture, and that is the diameter of the lumen. The size of the lumen, those arterioles, again, when they vasoconstrict and get really small, there's significantly increased resistance versus if they vasodilate and get very large, there's very little resistance, okay? The example there would be setting a fire hose next to a garden hose. If you put the same amount of water down each, there's going to be a lot less resistance through that fire hose than there is through a garden hose, right? It's significantly wider. All right, so from what I just talked about for the exam, know that, again, we've talked over and over about this, that the arterioles of all the blood vessel types, the arterioles are the major players, the major determinants of peripheral resistance and redistribution of blood. Arterioles steer blood to areas of need and away from areas where it's not needed, all with the intent of homeostasis and function, depending on what's going on at that given moment, okay? Also know what we've talked about related to this is that smooth muscle type tissue in the tunica media, the wall of the arterioles, when it relaxes, it's going to dilate, increasing the size of the lumen, which then would decrease resistance, okay? Third question, then we have one more slide and we'll get you guys out of here to start your weekend for some of you. So what is vasodilation? I've used the term over and over again. Is an example of vasodilation, capillaries that get smaller in extreme cold, veins that contain thickened blood, or smooth muscle relaxation of vessels causing expansion or a large an increase in the size of the lumen of that vessel. All right, and last slide. And this is kind of a preview. We talk about it here because it's related to blood pressure and blood vessels. But we're going to certainly do a much deeper dig into this when we talk about the kidneys and their influence on blood pressure. Our kidneys are the major regulatory source of adjusting blood pressure. It comes back to what I previewed earlier when we talked about capillary beds, how our kidneys are, for the most part, recycling centers. One of the major things that our kidneys are analyzing and deciding to keep or discard is fluid, okay? And so if our blood pressure is high and we have a lot of blood volume, in other words, a lot of blood plasma or a lot of water, our kidneys will say we have sufficient blood pressure and they will choose to eliminate the water as urine. Okay. However, if our blood pressure is low and needs an uptick in adjustment, our kidneys are going to sense that and recycle water back into the bloodstream, thereby increasing blood plasma volumes, thereby increasing blood pressure back to homeostasis normal and not eliminated as urine. That's called the direct renal mechanism. That's the first line of response to blood pressure variances, disruptions of homeostasis by our kidneys. Do they eliminate water because our blood pressure is too high, or do they reabsorb it because our blood pressure is too low? The second method that our kidneys work to manage blood pressure with is what's called the indirect mechanism. And this involves hormones. And these are hormones that we really didn't talk about at length in the endocrine system. If you remember when we talked about endocrine, I said that we were going to focus on the key primary hormones, the big ones like growth hormone and testosterone and glucose. Okay. Renin, angiotensin, aldosterone are kidney-related hormones that are significant and they're important to maintaining blood pressure, but they're not major hormones. But the renin, angiotensin, aldosterone system, really in a nutshell, is back to that analogy of playing basketball on a hot summer afternoon. Okay, we're sweating. What's the first thing that happens on a hot day or just even on a cold day if you're exercising? You get thirsty, right? Well, that is the release of hormones from the kidney that are going to trigger your hypothalamus in your midbrain to send a signal to your cerebrum to make you realize and can create the sensation of being thirsty. When you exercise, you get thirsty. So that is the kidneys releasing hormones that trigger the hypothalamus to tell your body and your brain, hey, I'm thirsty. You then take in water. And of course, when that water is taken in, it can get recycled during the direct renal mechanism and raise your blood pressure back up. Okay? In concert with that, at the same time, because it takes a little time for you to get thirsty enough to drink the water and then for its way to make it to the kidneys, right? So at the same time, the renin, angiotensin, aldosterone mechanism will also hormonally start vasoconstriction, tightening the walls of the lumen, which we said, remember, the number one method of increasing resistance is to vasoconstriction, to reduce the size of a lumen. Okay, so kind of a two-pronged attack from the hormones renin, angiotensin, aldosterone released by the kidneys. It's going to signal to make you thirsty, so you take in more fluid, so the direct renal mechanism can work. and at the same time, vasoconstrict or narrow those arterioles to temporarily increase blood pressure until that water ultimately gets on board. And again, we'll get more into that when we get to the urinary system. So, all right, guys. So as promised, that was a short but sweet one today, relatively short. You guys should have 3 extra credit questions to send to me. Drop those to me right away after class. And for the most part, if you guys are off, if this is the end of your week, have a tremendous weekend. Be safe. Enjoy this nice weather. Get out there and enjoy this beautiful afternoon. All right. Have a great weekend, everybody. We will see you guys on Tuesday when we start the respiratory.